Our investigations use a new animal model of Parkinson's disease, or PD, to determine how progressive reduction in dopamine production alters the function of striatal cells that produce acetylcholine. Cholinergic interneurons in the striatum fire using hyperpolarization activated cation or HCN channels that repolarize the cell after each action potential. They also express dopamine receptors that allow the cell to react to dopamine and encode motor learning. There is a reciprocal relationship between the two chemicals. Dopamine levels fall when acetylcholine levels rise. Researchers extended this relationship to PD, even though it had not been adequately investigated. The treatment of PD has primarily relied upon boosting dopamine using L-DOPA. In this study, we developed a mouse model of PD where dopamine-producing cells progressively decline. Unlike other models, dopamine levels in these mice progress similarly to humans with PD. Using behavioral, biochemical, and electrophysiological techniques, we found that availability in both neurotransmitters fell, but acetylcholine declined less than dopamine. Unlike normal mice, the dopamine-deficient animals demonstrated that dopamine availability promotes motor learning, while acetylcholine inhibited performance. This last finding about acetylcholine suggests that current PD treatment is inadequate. We found that the reduction in striatal acetylcholine stems from two changes in the cholinergic interneuron. First, the cells are unable to make a sufficient quantity of acetylcholine due to a reduction in RNA that is required to create choline acetyltransferase. Secondly, the cholinergic interneuron fires at a much slower rate due to a reduction in RNA that creates the HCN channels. This reduction in RNA continues to decline as the disease progresses. Dopamine receptors on cholinergic interneurons also adapt to the decline in dopamine. After acute dopamine depletion with reserpine, inhibitory D2 receptors are more active. In the Parkinson mice, D1 dopamine receptors are more reactive. To summarize, in normal mice, cholinergic interneurons have a reciprocal relationship with dopamine. Dopamine release reduces interneuron firing and acetylcholine release. In acute severe dopamine depletion, dopamine further reduces interneuron firing and acetylcholine release. In moderate progressive dopamine deficiency, acetylcholine production and interneuron firing are low. Dopamine released from any surviving axon boutons increases interneuron firing, and the acetylcholine dopamine ratio increases even further. The interneuron firing rate and acetylcholine production decreases with the length of disease. Therefore, the use of L-DOPA in advanced PD would enhance the imbalance between dopamine and acetylcholine, perhaps leading to dyskinesia. In conclusion, we hypothesize that a targeted approach to restore the proper neurotransmitter balance may improve clinical outcomes.